My name is Ponchanok Weeriyanun, and I will be presenting my thesis on the topic of assessing heat tolerance of adult corals sourced from across the Great Barrier Reef. So the coral reef, or the Scleractinian coral, is the invertebrate living form composed of clonal polyps. It can secrete the calcium carbonate to form the skeleton under its tissue. Hence, it creates the foundation of the coral reef. And in the coral tissue, there is the dinoflagellate symbiont called zooxanthellae, and the majority of the energy used in coral is gained from these microalgae. And when the symbiont enter the thermal stressful condition, the symbiosis relationship breakdowns, and coral will lose the symbiont cells or pigment, causing them to become pale, or as we know as the coral bleaching. Coral will lose its energy resource and die. And there are many responses to the increasing of water temperature, and one of them is local adaptation. There is a study of Keppel et al. In 2015 stated that juvenile inshore corals show different patterns in growth under heat stress. Another study of Oliver and Palombi in 2011 said that corals in a higher thermally variable pool had higher survival in a heated treatment. Finally, the study of Quigley et al. 2019 said that coral parents from warmer regions had transgenerational symbiont mechanism. There is also an evidence of coral adaptation in the Great Barrier Reef of Australia. This picture shows the mass bleaching events in the Great Barrier Reefs across the year of 2016, 2017, and 2020. You can see that the trend of bleaching showing in the red dots vary through years. So, the aim of this study is to assess the physiological responses of the coral Acropora tenuis to heat stress. And the hypothesis of this study is that coral fragments that located in the different locations along the Great Barrier Reef will perform differently in the physiological response to the exposure of heat stress. For the methodology, coral colonies were collected in November 2019 in 3 study sites. The first one is in Esk Reef, which is located in the most northern part of the Great Barrier Reef. It has monthly mean temperature equal to 28.8 degrees Celsius. Second study site is Davies Reef, which has the monthly mean temperature 28.4 degrees Celsius. And the last study site is Keppel Reef, which is located in the most southern part of the Great Barrier Reef. It has monthly mean temperature equals to 27.3 degrees Celsius. Then the coral colonies were brought back to National Sea Simulator or SeaSim at the Australian Institute of Marine Science. And for the fragmentation, 3 to 5 colonies were selected to represent the different genotype in one population. The one population here means the corals that collected from one study site. And for the fragmentation, coral colonies were fragmented into at least 6 branchlets. For the Keppel colony, we fragmented 28 fragments from 3 genotypes. Then we made 51 fragments from 3 genotypes of Esk as well. And for Davies, we made 82 fragments from 5 genotypes. After we get all the branchlets or the fragments. We put them randomly into 6 experimental tanks. We had 3 tanks for the hot treatment with 31 degrees Celsius temperature and another 3 tanks for ambient treatment with 27.5 degrees Celsius. The light intensity is 171 par with 12 and 12-hour schedule. We feed the coral daily with artemia 0.5 individual per milliliter and microalgae 2000 cells per milliliter. And for the physiological measuring, we measure the physiological responses of fragmented. First is the effective quantum yield, which represents the photosynthesis activity. We use the pulse amplitude modulation or PAM chlorophyll fluorometer. And for bleaching severity and necrosis, the image was taken using the digital camera, and then the color of the fragments was compared with the coral watch card. It can demonstrate the intensity of the bleaching. And the IMBRSea program was used to analyze the red, blue, green standard curve of the color of the fragments and the color in the card. The same program was also used to measure the area of the tissue necrosis. And finally, for the mortality, we observe the coral fragments and consider them dead when the microalgae cover appeared on the surface of coral. And for statistical analysis, all physiological analysis were carried out using R's program linear models implemented in the LME4 packages. So we test the models and we saw that there was no influence from tanks or sticks that we put randomly. And then the diagnostic part was used for the model assumption. And then we use the ANOVA function to calculate the p-value, and the pairwise test was used to assess the specific effect of temperature and population. And to assess the differences in survival, a binomial distribution linear effects models was used. So for the result, the survival was not significantly impacted by the population, but the high temperature affected the percentage of survivorship significantly with p-value 0.002961. As you can see that in the ambient temperature or in the blue square, the survivorship remain 100% in all 3 populations, but in the hot treatment, the survival of Esk is 84% and the survival of Davies is 95.12%. And for the percentage of necrosis, temperature and population had significantly impact on it. And when we run the multiple comparison of means in the population of hot treatment with 31 degrees Celsius temperature, we found that Esk and Keppel had significantly different percentage of necrosis. For the bleaching severity or the bleaching score, it was also significantly impacted by the population and temperature. And in the hot treatment, we ran the multiple comparisons of mean and found that Keppel's here and Davies had significantly different bleaching score. And finally, the result of effective quantum yield was also significantly impacted by the population and temperature. In the hot treatment, the effective quantum yield was highest on Keppel, followed by Davies and Esk. And after we ran the multiple comparison of mean, we found that the Esk and Davies fragments had significantly different effective quantum yield. So for the discussion from the result that we found in this study, we can see that from the physiological responses, they were different in the responses across the coral population in the Great Barrier Reef, as the result show that the coral fragments from Keppel Reefs had significantly lowest bleaching score and the percentage of necrosis. And on the other hand, the coral fragments from Esk Reef had significantly lowest effective quantum yield. So we can say that the Keppel's fragments had better fitness under the heat stress comparing to the fragments from Davies and Esk Reef. Even though the Keppel's had low thermal history, it might be because Keppel's reef is an inshore reef, which is quite not similar to Esk and Davies Reef. And there was also the difference in bleaching score, as we see that the Davies has highest bleaching score, or meaning that it bleached the most. However, the effective quantum yield was not significantly different among Davies and Keppel's Reef. Well, there might be some explanation that there was some adaptation in the Davies fragment that varies the photochemistry of the zooxanthellae, as it fit the result of the study of Parkinson et al. 2016 that said that the thermal history played an important role in shaping the community of the zooxanthellae. And finally, I'd like to discuss that the coral traits of fitness can be used as proxies of physiological fitness to be used for the reef restoration. Since there were previous studies observed many traits, such as measuring the skeletal growth rate via the buoyancy weight, or measuring sperm motility, or measuring the reproductive size. But in this study, we measure the effective quantum yield because the study of Skirring et al. 2018 stated that the measuring of effective quantum yield had benefit because this value is directly affected from the thermal stress, and the method of measuring is less time consuming, and also this value can be used to estimate the recovery potential of corals. And in conclusion of my study, I would like to say that the physiological responses of coral vary with historical temperature experience as well as the location. And the effective quantum yield, which represents the photosynthesis activity, could be used as an indicator for the reef health. And finally, choosing fragments with optimal fitness or optimal traits could help with reef restoration efforts. Finally, I would like to thank you, Australian Institute of Marine Science staff and members and the Assist Gene Flow team. I also would like to thank my family and friends, IMBRSea, Ghent University, European Union, and finally, thank you my supervisor, Dr. Kate Quigley, and thank you everyone for your attention.